My name is Amal Lahiani, and I'm doing my PhD between the Pathology Department of Roche and the Technical University of Munich. My poster is about stain virtualization in bright field imaging in digital pathology using an unsupervised deep learning approach. The goal of this method is to generate FAB-CK images from Ki67CD8 images, which is the first image there. And this would allow us to multiplex different stains on the same tissue and with the same coordinate system without having to deal with slide registration. And the use of an unsupervised method allowed us to avoid the need to generate and to deal with registration in order to create our training data set. So what we used is the cycle-consistent generative adversarial network, also called CycleGAN. And this network is built under the assumption that generating one image from the first domain to the second domain, then back from the second domain to the first domain would result in the same input image. So the objective function of this network consists of two types of losses. One adversarial loss forcing the network to generate images that are as similar as possible to the real images, and a cycle consistency loss minimizing the difference between the input image and the generated image obtained after cycle translation. Then, because of some memory limitations, the inference of the network was done tilewise. And this tilewise approach resulted in a tiling artifact in the reconstructed whole slide image because of the use of instance normalization layers in the CycleGAN architecture. For this, instead of using the classical inference, we propose to use an overlapping inference, meaning that we will use a smaller input size and a sliding window for the statistics of the instance normalization layer. And this allowed us to have a smooth transition in the statistics values, and it significantly reduced the tiling artifact. It's not very clear here in the image, but the image B there, this one, is obtained with the classical inference, and the image C here, it's obtained with the proposed approach. Then in order to validate our method, we generated 10 pairs of whole slide images with Ki67CD8 and FAB-CK images, and we generated the virtual images from the Ki67CD8 input images. Then visually, we noticed that CK reconstruction was more successful than the FAB reconstruction. And after discussion with our expert pathologist, we believe that this can be explained by the nature of FAB, which is a functional stain rather than an anatomical stain. Then for quantitative validation, we used an algorithm for CK and FAB cell detection. And we, as we can see here in this figure, the difference between real and virtual was smaller for CK than for FAB, which also consolidates our remarks about our visual remarks about these two stains. So we will investigate more this FAB reconstruction in order to hopefully improve its reconstruction. And thank you.
Thank you. Any questions? So you talked about this smooth transition in this statistic. If you can expand it a little bit, just what did you mean by that?
Yeah, so usually when we do the classical inference, we have these instance normalization layers, and then in the function that will be applied to the input images, we will have the mean and standard deviations of this input field of view in order to generate the output image. And when we have non-overlapping inference, then this transition in these statistics is huge, and then we get this tiling artifact. So we try to do step by step, do a smooth transition, and use a smaller input size. This is a little bit expensive computationally, but it helped us reduce the tiling artifact in the results. And also we are investigating this to try to solve the problem from the training part with changing these instance normalization layers with something more efficient. So it's one of the next steps.
Excellent. Let's thank our last speaker and all the speakers in the morning session.